All right, so I welcome all of you back after the break. We now have B. Anand. He's a professor at Indian Institute of Technology, Guwahati, which is in the eastern part of the country. Anand did his PhD, well, he did his BTech from Anna University, and then he also has an Bangalore/IISc connection where he spent a couple of years in the MBU department, then moved on to do his PhD from IIT Kanpur. And Anand's group has been interested in using structural computational bioinformatic tools to try and understand CRISPR-Cas adaptive immune system and also ribosome assembly. I would like to add that we are all here for an RNA meeting, but India has an RNA meeting of its own, known as the India RNA meeting, where several RNA biologists in India get together every 1.5 to 2 years. And the previous one, which was about a year ago, was conducted by Anand and Kusum here. It was a fantastic conference. So now we are looking forward to the next one, which will be conducted in Bangalore. So with that, Anand, it's all yours.
Thanks, Raj, for the generous introduction, and namaste to everyone. So this is the the campus that I work with, and it is basically nestled on the bank of mighty Brahmaputra. And apart from this, our campus, this part of the country has very interesting locations to explore, especially for the wildlife enthusiast. So I do invite you to explore this part of the country as well. Okay, so with this, my talk is going to revolve around this an interesting immune system that are very unique to prokaryotic systems. But it so happened that when I started my group, CRISPR was an obscurity, and it was yet to become a buzzword. So therefore, it so happened that our group evolved with the field, and therefore my first part of the talk is going to be mostly on the questions of what, when, and where I'm going to introduce the system with respect to the microbe interaction and the molecules that are involved there. My second part of the talk is going to talk about the mechanisms, where I'm going to address the question of how there. So in the interest of students and diverse audience here, my talk is going to be mostly pedagogical, especially the first part. So I hope you will enjoy that. Okay, so my group at IIT Guwahati is interested in the mechanistic aspects of RNA biology. Under this ambit, we focus on two problems. One is on the mechanism of ribosome biogenesis, especially in bacteria. The other is also a bacterial system, that is what is the biology and technology of the CRISPR-Cas system, this is also prokaryotic specific, which is also happened to be the focal point of my talk today. We address these two problems using a combination of approaches. So with respect to the CRISPR, before introducing that, let me introduce the microbes first. So all cellular organisms are predated by viruses, and therefore, bacteria and archaea are no exceptions to that. And therefore, in order to counter these infections, they have evolved with various kinds of defense mechanisms. And these defense mechanisms acts in sort of in several layers. And the first line of defense is something like very similar to how our skin protects from foreign invasions. And the first response actually starts from the surface. Typically, the phage recognizes a specific receptors on the surface of the bacteria, and they bind to these receptors and then inject their DNA inside. But in response to that, the bacteria actually can evolve with the mutations in the receptor, and therefore when a phage tries to recognize this, it can't, and therefore it will be excluded away. So this is called surface exclusion. So when this defense layer is broken, it triggers the next layer of defense, that is that widely known restriction modification system. So this layer actually gets activated when the phage gets inside the cell. So this is basically a nice defense mechanism that recognizes self from non-self. The methylation mark on the host DNA by the methylase acts as a self recognition mechanism. And therefore, the nucleases, whenever there is a methylation mark, it won't cleave the host DNA. But the phage, okay, so this is a methylation mark. So therefore, whenever there is a methylation mark present, the host DNA is not going to be cleaved, but the foreign DNA will not have this methylation mark, and therefore it will be cleared. So in this way, it is the second line of defense. But if this defense is also broken, you have a third defense that gets activated. That is more, sort of a very extreme defense. This is basically called as toxin-antitoxin systems. And these toxin-antitoxins are basically occur as a couple, so that the toxin activity is basically nullified by the presence of antitoxins. During the phage infection, this equilibrium gets perturbed, and therefore the toxins are liberated from the antitoxins. And once these toxins are liberated, basically they actually go haywire, and then they completely miss the entire system, and the cell dies. So you may ask, if the cell dies, why I'm calling this as a defense system? This defense system does not operate at the individual level. It's sort of an altruistic system, so where the bacteria sacrifices itself in order to basically limit the infection to the population, right? So these are some of the glimpses of the bacterial defense mechanism. So all of these have one common nature, that is they are all intrinsic in nature. They are innate defense mechanism, just like your skin, your tear glands. So these are all intrinsic defense mechanism that doesn't discriminate against the pathogen, and they respond to all kinds of infection, right? But we also have an advanced defense mechanism that is adaptive in nature. I don't have to tell you what is an adaptive immunity. We just came out of the COVID-19 pandemic. So, but very contrastingly, the prokaryotic world does not known to have an adaptive immune system. It is believed for a very long time that it is absent in the systems until the discovery of this RNA mediated immune system. So before, I'm sorry, actually, why this innate immune system is very interesting, not only from the point of defense, it is also from the point of our scientific activity, because the restriction modification systems can be repurposed. And basically, we all aware of what is a recombinant DNA technology. So in 1973, Herbert Boyer and Stanley Cohen actually devised a method that we refer to as molecular cloning today, and they developed this recombinant DNA technology. So therefore, this bacterial defense mechanism, though it is not only defense, it is basically a war between this bacteria and the bacteriophages. In a war, nobody wins, but as a bystander, we get to gain a lot of things in terms of concepts, ideas, and technology. Okay, one such glimpse is that recombinant DNA technology. So now coming back to this defense mechanism, so these are all intrinsic in nature. So we don't have an adaptive immune system in bacteria, and this notion was actually appended when indeed an RNA-mediated adaptive immunity was discovered, which is nothing but CRISPR-Cas, right? So now, for the past 1.5 decades, there is a lot has been done, understood about the system. So for a general interest, I'm going to basically summarize the current understanding that has basically come from contributions from several groups, including ours. So I'm going to summarize those things in a form of a cartoon. So for that, I'm going to take you to the microbial world, and we are going to witness how this microphage interactions pan out, right? This is a bacterial cell, and in this cell, the bacteria has a special locus called a CRISPR locus, and this is the locus, and this locus has a peculiar feature. CRISPR basically stands for clustered, regularly interspaced short palindromic repeats. The acronym itself captures what it is. Let me explain a bit. So this locus basically consists of a repeat, which is approximately, it's around average size is 30, but it is variable, the length is variable. But these repeats are actually direct repeats. That means if you write my name is Anand, it repeats periodically after some time. So these diamonds are all direct repeats. And between these diamonds, you have sequences which are variable, right? And then the second is the spacer. The variable regions are also of similar length, but the length also can vary, right? So, and then there is an upstream element called the leader, which harbors the regulatory element. So this basically, these three components, basically I refer to as CRISPR locus or CRISPR array interchangeably. But when I say this, what I meant is these three elements, right? So adjacent to this CRISPR locus, you also have a Cas operon. Cas stands for CRISPR associated genes. And this Cas basically encodes proteins of different kinds. And both the CRISPR locus and the Cas genes together constitute what we refer to as CRISPR-Cas system. Now, let's see how this system actually protects the bacteria. So when a phage infection happens, typically the phage wins over, but in the event of unsuccessful infection, the phage DNA is broken down by the host nucleases, and a fragment of this broken DNA is taken up by a set of proteins called Cas1 and Cas2, about which I'm going to talk about later in my second talk. So this will take a piece of it and they will introduce it in a polarized fashion in a region that is very proximal to the leader. So this is the leader that I am referring to. As you have seen that it is actually introducing it very proximal to the leader region. And this is interesting. What has actually happened is that the bacterium has now vaccinated itself, right? So this is just a vaccination stage. So you know that, but when we got vaccinated, we don't actually pass our immunity to our progenies. But here the vaccination, why it is because we actually have the memory in the form of antibodies, which is proteins. But here the infection memory is stored in the form of nucleic acid. And therefore this immunity, this memory is basically nucleic acid based, and therefore it is heritable. So therefore, whenever it produces progenies, it can faithfully actually pass on the immunity to all its progenies. So in that way, though it is bacterial immunity, it is far superior than what immunity that we have. So in that way, microbes does matter, right? So they are very advanced in terms of this kind of defense. So what happens is, now that the memory, infection memory is stored, what you have to do is you have to retrieve this memory. How are you going to retrieve it? And it is retrieved when this is basically transcribed. Okay, so it is retrieved in the form of RNA, and whenever it is this CRISPR locus is transcribed, it produces a pre-CRISPR transcript. Okay, so the entire unit is transcribed as a single unit, and then this has to be basically undergo a process called maturation. So what does that mean? So it means that another set of Cas proteins, they actually join together and form a complex, and they basically recognize a set of regions in the repeat. Okay, and once they recognize these repeats, they cleave this off, and now you can see that they form what is called as a ribonucleoprotein complex. So it has both protein and RNA here, and these RNP complexes acts as a surveillance system, right? So they are like sentinels. They scout for any foreign bodies. So whenever a reinfection occurs, so what happens is, now that this RNA that is now bound to this RNP surveillance complex, from this we call as a CRISPR RNA, right? At this point onwards, it is called as a CRISPR RNA, which is a matured form of the pre-CRISPR transcript. And the foreign bodies are basically recognized by complementarity with the CRISPR RNA, which we also called as CRISPR RNA, can be called as guide RNA in different contexts, but with respect to immunity, we call as a CRISPR RNA. So once the complementarity is established, there are two things here. The one is the complementarity, the other is the self versus non-self discrimination, about which I will describe a little later. So once this complementarity is established, it can actually recruit an effector nuclease. So this effector nuclease can be in cis or trans. So once this effector nuclease is recruited, it basically binds and cleaves this off. So now the infection is actually neutralized, right? So this is the overall view of the CRISPR interference. As I said, the highlight of this immune system is that in the case of higher organisms, you have this infection memory stored in the form of a protein, but here it is stored in the form of a nucleic acid, right? So therefore, it can basically, it is heritable, it is passed on to the next generation, And therefore, a recurrent infection, whenever it occurs, all the progenies are also immunized, right? So they can actually elicit a robust counterattack, right? So now once we understood this, the next is what we learned from this. So the important thing to learn about CRISPR-Cas with respect to this talk is that it is an RNA-guided adaptive and heritable immune system, right? So now there are 3 operational stages. Operationally, you can actually look at 3 different stages. The first is the immunization stage that we refer to as the adaptation stage, right? So this is the stage at which the infection memory is stored in the genome. And this involves 2 things. These 2 proteins called Cas1 and Cas2 that I refer to as adaptation complex from here onwards basically binds to the fragment that is cleaved off from the phage. It specifically binds to a triplet, right? So this is basically called as a protospacer adjacent motif. And I talked about the self versus non-self discrimination, and that mark is basically established at this stage itself. So whichever fragment that has this PAM sequence, only that fragment is specifically captured. And this is also equally important at the last stage for the target cleavage. So therefore, this basically captures this fragment and then it gets stored, right? So the next stage is the retrieval of this memory that we refer to as maturation. So at this stage, the CRISPR RNA as well as these Cas proteins basically form the RNP surveillance complex. Right? The third stage is where the action takes place. So now this effector complex now recognizes the target through the complementarity between the guide RNA and the target DNA. And then the effector nucleus cleaves that. So again, the cleavage has to be licensed by the presence of these PAM signatures in the target, right? So while acquisition also, it ensures that it takes only those fragments which has this PAM, while cleavage also, it ensures that once this complementarity is established, the next rule is that it also has to match the PAM in the target. So once these 2 stages are actually licensed, then the cleavage takes place. Otherwise, it doesn't, right? So therefore, it is also a very good self versus non-self discriminator, right? So now these are 3 operational stages. But because this battle between this phage and the bacterium is ongoing, and this struggle for existence between these 2 entities give rise to an innovative offense and defense strategies. And therefore, as I said, nobody wins in a war, but as a bystander, we get to have a lot of interesting flavors. The flavor is that the CRISPR-Cas systems, they are not sit as static entity, they also get diversified. And the way they get diversified is that, so during the past decade and a half, so we have, because of the genome mining, we got to know many different variants of the system, and there emerged a unifying theme by which this can be categorized. The major theme is that we found 2 categories. One is that in which the effector complex that I described actually consists of 2 models. One is a class 1. In the class 1 is that the effector complex is a multi-subunit effector complex. That means you have a single RNA, but the protein component is basically many proteins. Okay, so this also has been diversified into several variants. So there are 4 types here, and each of these types have subtypes. So therefore, you have several CRISPR variants. And the other system is the class 2 system. So here, the effector complex is a single effector complex. And this effector complex have, again, 3 subtypes. And the salient feature is that you have 1 CRISPR RNA and 1 effector complex, 1 effector protein, right? So these 2, basically, here I have simplified things, but in actuality, we have about 30 different subtypes, and it is still counting. So the CRISPR-Cas systems are expanding as and when we get to discover more of these from the new metagenomics and things like that, right? So with respect to the cutting efficiency, if you compare that, the class 1 system is like a coarse cutter, okay? But the class 2 system is like a Swiss knife, right? So you can basically see a Swiss knife has multiple purposes. You can slice it finely. You can also have a screwdriver that you can tweak it.
So there are many such purposes. It is a multi-purpose thing. So therefore, the class 2 systems are the ones that are majorly exploited for the biotechnological applications. Whereas from the immunological point of view, the class 1 system is the one that is very advantageous for the bacterium, because here it is a survival issue. Okay, I don't want to finely slice my enemy. I want to really kill him, right? So therefore, this is very, with respect to this purpose, the class 1 systems are highly prevalent. Almost 2/3 of the systems that we know of today belongs to class 1, right? Okay, so what kind of targets they actually recognize? With respect to the nature of the targets, it can recognize DNA and cleave that. It can recognize RNA also. And not only that, there is also an RNA-dependent protein targeting, right? So we also have protein targeting CRISPRs. So therefore, in terms of targeting, it can recognize DNA, RNA, and protein, right? So the class 1 and class 2 that I have described, I have already described. Class 1 is a multi-subunit effector, and class 2 is a single subunit effector. But you can see them, all of them can either target all 3 of these, right? Okay, so the next thing is, how do the systems evolve? Remember, we don't have a CRISPR system, okay, in our cells, but we have a similar cousin that is basically the RNAi, right? So the RNAi, with respect to the target recognition cleavage, is functionally analogous, but mechanistically it is very distinct, right? So the CRISPR-Cas systems, how it actually came into being. So we have a general hypothesis. What I'm representing is not a dogma. It is only a hypothesis, so it is going to evolve with time. So the hypothesis is that we have an ancestral gene that encodes for a ferredoxin like an RRM fold. See, with this RNA community, I don't have to tell what is an RRM fold. So you have a primitive RRM fold that basically got diversified by duplication and rearrangements over several generations. And therefore, you start off with a pre-ancestral RNA fold, and that got duplicated and diversified and gave rise to a series of Cas proteins. And all of these Cas proteins of the type of class 1 systems have these RRMs. We also call them as repeat associated mysterious proteins. The reason being is that we never knew that it was basically an RRM fold until we basically solved the structure. And it is not a canonical RRM fold that we all aware of. So there are a lot of insertions, rearrangements, and things like that. Therefore, it is very difficult to identify them from the sequence level. So once these Cas genes have expanded, only at this stage, the adaptation module has been acquired. So it is basically acquired from the transposase. So the transposons, you know, they are mobile genetic elements. And I talked about 2 proteins, Cas1 and Cas2. The Cas1 is basically a transposon. Its ancestor is basically a transposon that we call as a Casposon. And the Casposon and the Cas2 is in CRISPR, it is an inert protein, but it is actually a part of toxin-antitoxin system, and it is a toxin. But when it actually been acquired in the CRISPR, it lost its activity, so it is no more toxic. So the Cas1 and Cas2 adaptation module was acquired from the transposons, and then this became the first ancestral CRISPR systems, which got diversified further and gave rise to all kinds of flavors that we know today, right? So now, what is the theme that we understood from this evolution is that the class 1 systems show a monophyletic evolution. What I mean is that you can trace this evolution of all these to a common ancestor. So they evolved by monophylogeny. But with respect to class 2 systems, there is for example, here we have 3 kinds. The Cas9, Cas12, and Cas13, they belong to type 2, type 6, and type 5. So the Cas13 system actually evolved from an HEPN, that is higher eukaryotes and prokaryotes nucleus. So this basically got duplicated, and then it has acquired the adaptation module independently from the transposons, and that is how the Cas13 system evolved. And the ancestor of this Cas13 is basically a family called ISCB, right? So this is again a transposase. And with respect to Cas9, it is basically evolved from another transposase family, which is called as TNPP. So this is all these three systems, and the Cas13 basically evolved from a toxin family. So all three of them basically have some connections to the transposase or a toxin systems. So the difference is that the class 2 systems have evolved not as a monophylogenetic system. That means they don't have a common ancestor, and each of them have independently evolved at different points in time, right? So this is with respect to that evolution of how the systems came into being. So now coming back to the CRISPR systems now. So as I told you that this is a very coarse cutter and this is a fine cutter, and therefore we can basically, with respect to the biotechnological point of view, the class 2 systems are the ones that are basically exploited. And the first system that was exploited belongs to this type 2 category that is called the Cas9. Many of you might have heard about it or are already using it. So I will just take this as a prototype and describe some of the concepts that we can learn from it. Okay, so this is how the Cas9 looks at it. So you don't have to look at the maze of information that is around here. Let me simplify that. So it is basically a bilobed structure. It has 2 lobes, okay? The lobe 1, which we call as a recognition lobe, that recognizes the binds to this guide RNA and basically recognizes helps in the recognition of the target. The other one is the nuclease lobe that harbors the nuclease domain. So there are 2 nucleases here, the RAUC as well as the HNH. Let me basically have a panoramic view of how it looks like. So the color coding is basically the same. The RAUC domains are not a continuous domain. It is basically a discontinuous domain. So, but in the 3D, it basically becomes a continuous structure. So now, if you look at it, so you have the red ones are the guide RNA. So in the case of the Cas9, we have 2 kinds of RNA here. Okay, so let me first describe this. This is basically the RAUC. This is the HNH, and then you have the recognition lobe, and you have the PAM interacting motif. Okay, so this is basically, it has a 2 nuclease domain now. So let's simplify this further, and then we can see how it basically recognizes the target. Okay, so when you have a genomic DNA, so when you have reprogrammed this Cas9 system, so your guide RNA basically has 2 kinds of RNA. One is the CRISPR RNA that I have already described it, but very strangely to the system, it also has another RNA which has an antisense to this CRISPR RNA. So therefore, both these RNAs are important. So both the sense, that is the CRISPR RNA and the antisense RNA, basically forms a base pair, and this base pair is processed further by the RNase 3 within the host to form the mature CRISPR RNA. And therefore, and both these RNAs have to be there either in trans or in cis, but it has been repurposed in such a way that you don't have to express them in trans. It has been stitched together through a covalent bond, and therefore it is called as a single guide RNA. Okay, so this is very specific for the systems, but some Cas12 systems also has a tracer RNA that you have to repurpose it. So now the Cas9 systems, once it actually recognizes the target, so it base pairs with the target, and then once it base pairs, in order for the cleavage to occur, as I told you, there are 2 things should actually occur. One is the complementarity between the guide RNA and the target. The other one is the self versus non-self recognition, right? So in the case of the Cas9 systems, the PAM motif, which is basically a triplet, NGG, and this triplet is located in the distal side, that is at the 3' end, okay? But most of the other CRISPR systems, you will find these PAM motif at the 5' end, right? So once these PAM recognition also happens through the PAM recognition module, so then the nucleases basically can cleave this. So the HNH cleaves the target strand, yeah, and the RAUC cleaves the non-target strand. So the cleavage can occur. What happens here, the another thing to note here is, Once the target recognition happens, the DNA gets unzipped, right? And then you have, this is what I refer to as a target strand, which base pairs with the guide RNA. This is a non-target strand, and this structure is also called as a OR loop, right? So whenever there is base pairing recognition occurs, there is an OR loop, there is a bulging that is happening there. Okay, so once the cleavage occurs, what happens? Now the system is under a genomic insult. So a source response is triggered, right? So all the kinds of repair machineries are now got to work, and the rest of the action is not by the CRISPR systems, it is basically by the host systems, host repair machinery itself. So now you have a genomic insult, which is a lesion. This lesion has to be repaired. So what are the different means by which these lesions can be repaired? So one way is like you simply seal them, okay, and that is what is performed by this repair mechanism called as non-homologous end-joining repair, okay. So though it is fairly good, but it is also error prone, right? So when it actually ligates the cleaved end, there are different possibilities could occur. The first possibility is that it can seal it perfectly without any errors. So in that case, you get back the wild type, right? But it can also do indel operations. It can either insert something or it can also delete something, right? So these indel operations, and if it happens in the part of a region where the coding sequence is present, then it can also probably lead to frame shift, right? So therefore, you can basically, the functionality of that particular open reading frame or the gene is going to be affected, right? So this is, these are the things that happen. So these are the possibilities that happen. So here, all the indel operations that this NHEJ repair machinery does it, you don't have any control over that, right? So you can't predict it, you can't have no control over that. So, but it is something of a random chance that you have to wait for, right? The other kind of repair machinery that can be activated requires a template, right? So this template is basically called as, should have a homologous region to the target site. Therefore, whenever you have this template as an external DNA, which we call as a donor DNA, then this HDR machinery can use this homologous region, and using this double crossover can basically repair this end. So by this process, you can also introduce a region which is external to the genomic DNA into the genome, right? So this way you can basically do a knocking, right? So these two kinds of repair machinery. So whenever you want to do, so all these steps, operations that I described is very similar to what a kind of a word processing that we do, right? Indel deletions and things like that. So therefore, it is also called as editing. And when you edit this at the level of gene, it's called gene editing. But if you do it at the scale, at the genomic level, it's also called as genomic editing, right? So either of these two, if you have a donor DNA, you can actually insert this. If you don't have, then NHEJ can actually leads to the repair. Okay, so this is basically what I have described. There are two pathways that can majorly operate. Now, because it is very simplified, right? So all you have to do is you have to get this guy to the target site, and then the rest of the job is done by the repair mechanisms. So therefore, all of the living cells, most of the living cells have these repair mechanisms. So therefore, you can basically, in principle, adapt the system to do genome editing in any organisms. And this is a very simplified scheme. So therefore, this, in principle, this technology can be adapted for all living organisms, but the efficiency, as I just described, it is not only the CRISPR system that does the job, the repair machinery also has to co-opt, right? So therefore, different organisms have different repair machinery of different kinds. So depending on that, it will be easy in some, it may be difficult in some, right? So that is the challenge that you have. So the challenge is not to introduce, not in the CRISPR systems per se. The challenge is, with respect to all these organisms, you have to introduce the CRISPR machinery because none of them encode an endogenous system, right? So you have to heterologously introduce that. So therefore, you have to find a way to deliver the system to the target sites with high fidelity. And then, the repair systems also have to work efficiently for the edit to happen. Okay, so now, see, this is basically a kind of a genetic scissor. We also had a genetic scissor that I described in my initial part of the slide, which is nothing but which led to the development of the recombinant DNA technology, right? So the restriction endonucleases. So we have this restriction endonucleases for several decades, but why it didn't take off to that level that we see for the CRISPR is the reason is that the restriction endonucleases recognizes a selective sequence which has to be present in the genome. So if it is present in the genome, we can use that to cut, right? But if it doesn't, you cannot do, right? So therefore, your capacity to edit a particular genome depends upon the random chance by which these restriction endonuclease sequences are present, right? But with respect to CRISPR, and that is the same fate with zinc finger nucleases and talents, which are the predecessors of CRISPRs, right? So they also have met the same fate. But the beauty of CRISPR is that because it is programmable, just by simply changing the sequence of the guide RNA, you can target it to this part of the genome or to the other part of the genome. And you can also do it at level of, genome level at different scales, at higher scales. So therefore, it is also, it is scalable, and you can also do multiplexing where you can have multiple guide RNAs you can have, and you target a different location at the same time. So all these are possible just by changing the sequence of the guide RNA. So therefore, the CRISPR is much more versatile compared to the predecessor genetic scissors. Okay, so we are not only, so CRISPR is a good research tool, but that is not what actually got the excitement going, right? The excitement is that we know that by just using this genetic scissors, can be able to apply them in a clinical context, and that's what it is all about. So because I told you that it can be repurposed, we have a lot of genetic diseases which are basically because of the point mutations, and can we use this genome editing or to correct that diseases? So one such thing that I just want to describe here is that sickle cell anemia. For this audience, I don't have to describe this, what a disease is. It is because of the point mutation, which leads to a normal hemoglobin to become a sticky hemoglobin, and therefore that changes the phenotype of the RBC, which is also fatal if it is homozygous, right? So now, what we can actually do, which was also demonstrated, is that we can take the HSPCs from the patient, and in vitro you can edit it, right? And you can transplant again. So therefore, the cells can now produce a wild type gene, and therefore you can cure this disease, right? So it is easier said than done, but in this case you can actually do an editing in vitro, right? But many cases we may not be able to. For some diseases, we have to introduce the CRISPR systems inside the body, and that's where the challenges that I described earlier comes into play, right? You need to have a delivery vehicle which is non-toxic, precise, and should not have any other side effects, right? So that is a challenge. But it is also encouraging that in recent times that even US FDA has given approval for some of the treatments using CRISPRs, right? So because this impact is in a very short span of time, the impact is widely felt, and in recognition of that, both the Emmanuel Charpentier and Jennifer Doudna in 2012, they repurposed this the single guide RNA and showed that you can edit the genome. And in recognition of that, in 2020, they won the Nobel Prize. But these two are they are not the only ones who developed it, but we have other heroes of CRISPR who developed it. But anyway, but they represent the face of CRISPR. Okay. So now, in addition to what I described as a Cas9, you can also take this to a next level, and that's what David Liu's group at Harvard has done it. So typically when you use Cas9, you have to introduce a double stranded cut, right? And also you don't have a control about the editing, and therefore can this be simplified? So in this case, the David Liu's group actually made some sort of an editing. That's what I said. It's like a Swiss knife. You can't win it. So here they have stitched this Cas9 with the cytidine deaminase. And then they have mutated this nuclease. So it's basically, essentially, the nuclease activity is not there. So here the Cas9, which is basically a dead Cas9, can recognize the target, but it cannot cleave it. right? So here you are using it as a delivery vehicle to localize to a particular target site. So once you localize it, suppose the Cas9 binding region leads to this R loop, and this is the PAM site, right? From the PAM site, most of the regions are covered by Cas9, and if distal from the PAM site, you have some, right, which is single-stranded. And the cytidine deaminase actually acts in a single-stranded DNA. So therefore, if you want to correct this, what the cytidine deaminase does is that it basically oxidizes this amino group. So once you oxidize it, you convert C to U, and therefore this C can, can be converted to U, and this U is basically not native to DNA, and therefore your repair machinery basically clicks in. So uracil DNA glycosylase recognizes and removes it in normal circumstances, but you also have another inhibitor protein attached to it, so it cannot operate on it. So therefore this stays on, and during the replication cycle, it basically gets switched to TA pair. So therefore you can also do the, this is basically a CG editing. You can also do an AT to CG editing if you, instead of a cytidine, if you use an adenosine deaminase, right? So this is basically a single base pair editing. You can also advance it further. So this is a base, this is called as a base editor. So you also have a prime editor. So prime editor is another advanced version. So where you don't edit as single base level, so you take it to a new whole level. So here what you do is that you extend this single guide RNA longer to have a template like this. The red ones are the template. This is called as a prime editing guide RNA, right? So it's longer than what a single guide RNA is. And here you selectively neutralize the HNH. So HNH is dead, but the RevC is active, right? So the active RevC can introduce, and here to this Cas9, you stitch a reverse transcriptase. So you can actually introduce a nick here because only RevC is active. So once the nick is once a nick is introduced, you have a 3' end which can be extended by the reverse transcriptase using this RNA as a template, right? So you have extended this RNA, so it can act as a template. So it basically, if this is the region that you want to introduce, you can basically introduce that because it can act as a template for the reverse transcriptase. But this 5' flap, which is also redundant with respect to this, will remain, and it can be get rid of by the repair systems. So that in this way, you don't actually have constraints that the base editors have. So the prime editors, with prime editors, you don't have a constraint. You can basically extend the region, right, without any constraint, and basically it depends on the host systems and the efficiency there. But all these systems that I described, conceptually, in principle, it's doable, but in practice, you have to basically optimize with respect to the respective systems, right? So this is how an overview. So this is an overview of the CRISPR system as an immune system. By understanding about the basic functional mechanism of the system, this is a good demonstration of how this can be exploited for other biotechnological and clinical applications. Right. So with this, we can have a break, and then yeah. So I'll just.
Yeah. Thanks, Anand. Do you want to take a breather, or we can have question and answers? Okay. Wonderful. Yes.
Hey, Anand, nice talk. So I want to ask regarding self versus non-self, right? You said there is only 3 motifs for self, right? Most of the systems. So do you think only 3 nucleotides gives this self versus non-self, or there are other mechanism also?
Yeah, that's a good question. So the question is whether I mentioned about 3 triplets for the self versus non-self recognition. Because it is very short, the question is whether it is going to be effective, right? What we saw is if there is a correlation between the length of the triplet as a PAM versus the size of the recognition motif, right? If the triplet size is less, the recognition motifs are enlarged. Okay, it's because the triplet size is less, so the sensitivity is less, so it has to be enlarged. So the recognition motifs are expanded, and in order to make the recognition even more sensitive. Whenever the triplet, whenever the SPAM motifs are expanded, we see that the size of the Cas system itself is small, right? So the sensitivity is basically switched between the nucleic acid and the protein in that way.
So Anand, this CRISPR only makes a cut, correct? But when it is released from the DNA, it can make a cut for long sites. Then how can you call it as precise edits? Can it incorporate? If suppose I want a one event, right? If I want a one event,
okay,
if it is breaking a lot of nucleotides, how can it make a precise edits?
Okay, see, there are, if you are using a class 1 system that I described it, it's just like a handsaw, right? So it's a very coarse cut, you can't call it as a precise. So that is evolved as basically a way to protect the bacterium, right? Sporadically, the Cas2 systems are sporadic systems and they are very...
No, but I'm limited to Cas9. What globally is using Cas9 system?
Yeah, but the cleavage is very precise, right? It happens at the place you are...
That is only cut. But repair happens through the endogenous systems.
True,
endogenous
that's what I
systems,
said.
right?
That's what I said.
Yeah.
That's what I said. You don't have a control about the edit.
Yeah, exactly.
You don't have a control because it is not only the CRISPR system, it is the host repair machinery has to work in coherence. So
what recruits?
Yeah.
What recruits? Whether Cas9 has any activity to recruit those repair enzymes on the DNA?
Absolutely not, because
No, right?
see, all these Cas systems are external to the host. So they don't have any crosstalks like that. So it is recognized by because the double-stranded break is basically a lethal deletion. So it sends a source response immediately and you have a repair machinery recruited to that location and that does the repair job. Right.
Another question regarding your PEG, prime editing, right? So when you're linking Cas9 with reverse transcriptase, they're basically very not mobile, right? Or is it linked to a longer linker so that it can go to the different places on the cut site?
If you look at
so
that.
What is the distance between Cas9 linker domain and the reverse transcriptase that is attached with it?
Right. See, it has to be, this should be an optimum length. In such a way, it should be like, it should not be very constrained and it should be it should not be too flexible. Otherwise, it becomes very floppy. You cannot have. So it basically an optimum, but the exact length I don't have. I have no idea now, as I couldn't recollect that. But it should be an optimum length that should ensure that it should not be too close because then it will affect the activity of this cleavage, right? It should not be too far away so that, it may not be able to come back.
Adaptive immune system in prokaryotes and eukaryotes. So in prokaryotes, you have the effector molecule, which is a nucleic acid, right? Which can be passed down to the progeny. But in eukaryotes, where the effective molecule is a protein, you lose that fitness advantage in terms of the species, right?
Yes.
So why do you think the adaptive immune system might have evolved in such a way that you lose that fitness advantage when you go from nucleic acids to proteins?
Yeah, that's an evolution, right? So we all evolved from an RNA world. And so it was earlier an RNA world and then now it is basically a combination of both RNA protein and then probably we are moving towards more protein world or something like that. I don't know. But the thing is that that's how it is evolved. So if you listen to Ramesh talk, he talked about the piRNA, right? So that's what I said. It's basically sort of a functionally analogous to that. So there is another kind of a kingdom called archaea, and in archaea there is something called an Ascard archaea, right? So it is also touted to be the progenitor cell for the earliest form of eukaryotic cell. So Ascard has CRISPR systems, right? So once this symbiotic event has occurred, somehow, I don't know why, it is probably, this CRISPR, the Cas proteins are all probably last, but the repeats we do have. That's what the piRNA that he described, they are still repeats. But there they target the mobile genetic elements. So CRISPR is also basically targets the mobile genetic elements. For example, phages and plasmids are nothing but mobile genetic elements. But here it is basically probably undergone some modifications. And by gene loss, we have lost all the Cas genes, but we have remnants of all those repeats still in our genome. Yeah.
Hi, I'm just a little curious. So if a bacteriophage is infecting a bacterium, and so you have the bacterium incorporating the infection memory, but say a bacterium is infected by hundreds and hundreds of different bacteriophages. So how feasible is this continuous incorporation of infection memory, like this genome expansion that we are seeing? Is it a reality? Is it possible?
Yeah, good question. The expansion, in principle, there is no limit for how far the expansion can go, right? But there is also some mechanism that takes place. That is, the acquisition happens in a polarized fashion, okay? And then the newly acquired infection memory happens at the one end of the CRISPR array. And so it gets added. So slowly the newer one become an older one and things like that when it goes to the other end. So therefore the selection pressure is always at one end. Okay, so the other end is like probably doesn't have such extent of selection, and therefore they can also degenerate. So the length of the CRISPR array.
You mean like an infection memory can be lost?
Yes.
Eventually.
If there is no selection pressure, if you don't have the respective phages in the environment, it can be degenerating as well. So it is basically an equilibrium between what kind of memory should be retained based on the selection pressure. If you have all kinds of phages that it is against which it already has a memory, and they are all the time around in the milieu, it has the selection pressure to retain that. But if it doesn't, it gets degenerated as well. So it is a balance. Okay, so the question of that is basically, so you don't basically need an external energy to cleave that, because the passport ester bond itself is, it's a high energy bond. All you need is, see, if you don't have an enzymatic catalysis, they themselves gets cleaved out, but it takes a longer time. But the enzyme just facilitates it faster. It brings the reactive group in such a way that the geometry is favored for the inline nucleophilic attack, and it basically activates that. So you don't need an external energy for that. It's already under higher energy. Okay. The second part of my talk is mostly from the work that we have done. So here I'm going to address the question of how, right? And mostly to deal with the mechanisms. And most of you probably have some familiarity with respect to genome editing and things like that. So some of you might have used it. But my emphasis is going to introduce you as an immune system. So I'm going to deal with how the bacteria actually recognize these foreign genetic elements and nullifies it. Okay. So just to recapitulate, I described about the 3 operational stages. One is the adaptation stage, where you acquire the infection memory. And the second is the maturation stage, where you retrieve the memory in the form of RNA. The third is the targeting stage, where you recognize the target and cleave it precisely, right? So now the hallmark of this CRISPR-Cas system is the first stage. That's an adaptation, because it's an adaptive immunity, right? So how it basically acquires the foreign genetic element and is the question that is the hallmark of this field. And we also got intrigued by this. So in the next few minutes, I'm going to describe about that mechanism and the work that we have done. Okay. So the acquisition, when it comes to acquisition of the foreign genetic element, there are 2 concepts or 2 aspects to it. One is the infected phage DNA has to be acquired by this adaptation complex, that is the Cas1-2 complex. It has to selectively acquire, okay? It has to capture. And I said that there is something called a PAM, which is important for the self versus non-self recognition. It has to be selectively acquired. And therefore, the first stage is about how it selectively acquires that, right? So that is how it captures. The second one is, once it acquires this, it has to basically install it in a polarized fashion, right? In a region that is proximal to the leader, right? So this always happens in a very polarized manner. Now you see, once it got, this piece is basically installed in a proximity to the leader region, right? So how it basically inserts it in a polarized manner with respect to the proximal leader is another facet to this, right? So we wanted to address this question. And this is also biologically important, the polarized addition, as Som has asked. So you see that, so because the newer spacers are always added at this end, leader proximal end, and so whenever the newer spacers are added, it is going to push towards the 3' end, right? So therefore, what this newer spacer actually tells you, it tells you that that is the current thread that is present. That is the current phage that is present now, right? So therefore, but the ones that are far away from this actually represents the infection memory that is probably not there existing, right? So therefore, this part of the memory can go on and expand, but it can also degenerate. But this part of the memory is always under selection pressure. Okay. So now let's describe the questions that we were interested in. So whenever, if you look at this, if this is the phage DNA, and this is the fragment that has been acquired, the adaptation complex, if after this phage DNA got cleaved, that is the boxed portion, it looks for the fragment. It can bind to any fragment. So it is not specific for this part of the N, the region that I have indicated as N, right? So it doesn't have any discriminated power. It can bind specifically to this part. But the discrimination part comes here. So it should have, the fragment should have this portion, right? So because that is very important. Otherwise, at the targeting stage, you can't have a self versus non-self discrimination, and it is going to be highly lethal for the cell. Okay, so this is an interesting part. And in the case of the system, one CRISPR variant that we were interested in, that is in E. coli, this is always, the size is always 33 base pair, very unique. So, and once it actually got processed, it basically cleaves it, and a part of the triplet is actually there in the infection memory. It is also added as a spacer, right? So we basically got intrigued by this. So we asked the question, what determines this, right? So this is what we are basically interested in. So the second question that we were interested in it, once it captures the spacer, how it specifically inserts them in the leader proximal end, right? So what determines this specificity? So these are the two questions that we were interested in. And these two questions were addressed by a talented PhD student, Anand. So I'm going to describe how he went about doing that. Okay, so the thing is that in most of the CRISPR variants that were known, this job of recognizing this PAM happens to be in trans. Okay, so this is the adaptation complex. It's like a butterfly. Okay, where there are 4 Cas1 subunits, and in the middle that you have a dimer of Cas2. So this recognition of the PAM happens in trans by another protein called Cas4. It recognizes the PAM. When you don't have this Cas4, therefore it means that you can't have a PAM recognition happening, right? So once it recognizes the PAM, it cleaves this, and therefore, you can actually, this Cas1 adaptation complex can actually uptake this, right? So when you don't have this PAM, so basically that means this process is blocked. But CRISPR is a system that is highly dynamic. It never, the CRISPR specializes in one thing, that there is no rule. You can't have a rule to be fixed, right? So I talked about this classification scheme. In the class 1 system, the most predominant one is the type 1 system. It has 7 subcategories. If you look at all of them, almost all of them have this Cas4 protein. So that means they already have a recognition system to recognize PAM in trans. But as I said, CRISPR is also very exceptional. It always throws up some surprises. So there is one system that is there in E. coli. It's a representative in E. coli. That is 1E system in which you don't find this Cas4, right? So therefore, in the absence of Cas4, how this system, this variant acquires the PAM, a PAM containing fragment is the question. So this is how the structure looks like. As I said, it's a butterfly. So where you have this pre-spacer is basically acquired. In the case of E. coli, it is 33 base pair, right? So the question is, the structure gave one clue that the architecture of the structure is such that it can basically retain only 33 base pairs, right? So that is one clue that the structure gives. But what it doesn't give is that whether it captures only those fragments with 33 base pair length, or it captures a longer fragment and then further it gets pruned into a 33 base pair. But the problem is there was a report that people have not shown that basically this Cas1 in E. coli can also process like Cas4 in other systems at the PAM region, right? So, but when we tested, we didn't find that to be the case. So therefore, also we came up with a hypothesis that perhaps the Cas1 can recognize the PAM by itself, and the host exonucleases may be pruning that. So in order to test that, we took a 63 base pre-spacer, and so this is the pre-spacer that is 63 base pairs. And when you actually provide this, Cas1 and 2 alone, you don't see any cleavage happening, right? So I don't know where it is. Yeah, here. So when you have only Cas1 and 2, it doesn't cleave, okay? It doesn't cleave. But when you have Cas1 and 2 in the presence of the exonucleases, you can actually see that there is a fragment that gets enriched and it closer to 33 base pair. So therefore, this actually tells us that in the presence of Cas adaptation complex, the adaptation complex basically protects this region from the host exonucleases, and the production gives the size selectivity, right? So then the thing is that this system does not have PAM. So then how it is going to basically selectively identify the PAM containing fragments? So that was the question. The structure also gave a clue to this. What we found was the CTD, the C-terminal terminal region of one of a subunit in the crystal structure. In the upper form, in the absence of DNA, it is actually it is in an extended form, and the extended conformation is basically stabilized by interactions between the amino acids. And you can see that this I291 comes from the extended tail, and this gets tucked away nicely between these two tryptophans in the Cas2. But when you have the DNA bound, this extended C-terminal tail is structured, and it is basically structured around the PAM containing region, right? So it interacts with the PAM. So this captured our attention, and therefore we hypothesized that perhaps the recognition element for PAM is sandwiched within the adaptation complex in the type 1E system. So how to test that? So when we analyze the length of other C-terminal tail of Cas1 in other systems, most often they were all around 12 amino acids. But only in type 1E, you can see that the length is almost double, suggesting that perhaps it has a recognition element. So in order to test that, we deleted the C-terminal tail. We also had penta point mutants and tested whether using an assay system, whether it specifically acquires the PAM. Okay, the assay system involves that you grow the bacterium for several generations and then check whether in the locus whether it has acquired the new spacers or not, and then you sequence that locus to identify whether it has any PAM. Right. So what I'm describing here is that in the you have the spacer sequences. So in the wild type the cognate PAM is basically AAG, but when you mutate the C-terminal tail or the pentamutant, the specificity for the PAM is gone, right? Suggesting that perhaps this C-terminal tail has the specificity element. Now, so what is that we have actually understood here is, so the Cas1 and 2 basically recognizes the fragment as well as the PAM by itself, while the Cas nucleases basically cleaves it off, and prunes the region such that it basically products only the 33 base pair fragment. So now the pre-spacer is captured. The next question is how it is integrated, right? So the integration is basically, as I mentioned, inside the cell, it happens in a polarized fashion, always to the leader proximal end. But if you do the same integration reaction inside the test tube, it is highly non-specific. That means it can randomly happen at any locations, right? So this immediately points to the fact that there is a specificity factor inside the cell that is dictating how it should, where it should go, right? So now the question is, what is that specificity factor, right? So the problem here is that how do you actually identify that specificity factor? The problem is you have, in E. coli, you have about 4,500 proteins, and I have to probably look for a handful of them to basically identify the specificity factor, which is basically looking for needle in a hashtag. So that this is, but here, in order to address this question, on a question on the CRISPR systems, we basically repurpose the CRISPR system itself to understand this question. So for that, we took this Cas9 system. So in this system, we have basically mutated the nuclease. So therefore, this is a dead Cas9 system. And we have added a 3x plaque tag, right? So this Cas system can localize to the target site, can be localized by reprogramming the guide. And this is basically acts like a purification tag. Now, what we have basically done is using this system, we can localize this dCas9 system to the leader proximal end. Because since the reaction basically happens towards the leader proximal end, the chemistry is somewhere happening here. So we basically want to pull out only those fragments that are here, right? So that can be done. So what we have in effect done it, this is for the information for the students. So if you basically want to identify such factors, you assume that this is where the Cas1 and 2 basically binds near the leader end, and you have an unknown factor that is bound here. So assume that I have localized the dCas9 here, right? So I can do an in vivo cross-linking experiment, cross-linking, so that things get cross-linked, and then you can break open the cell, fragment the DNA, and now these are all noises, right? So other parts of the fragments are all noises. So therefore, using this tag that is I have on the dCas9, I can fish out only this portion, and I can do a mass spec to find out what are the factors that are actually bound in this locus. So when we did that, as anticipated, we also found Cas1 and 2. In addition to that, we found another protein called integration host factor, right? So and others, right? So these are all called nucleotide associated proteins, right? So so we basically immediately we can associate what we are looking for, because we are looking for a factor that should have some role in integration, right? So the integration host factor is already performing that function with respect to phage integration, but it also came as in our screens. So the integration factor also belongs to a family of transposase, and it helps with the integration of the phage. So it has two subunits, alpha and beta. So the next question is whether this can be our candidate or not. So how do we test it? So we can basically test it using another simple assay. This is also for students if you are interested. So basically you can look at the, you can basically screen for the acquisition event by using a simple PCR experiment. So your spacers are all unique, so that you can have one primer targeting here and one on the leader region. So you will get a defined fragment. And if there is no expression or no expansion of this CRISPR, then it is going to be a constant fragment. But if there is an expansion, if you have a primers here, then you will get a multiple of 60. Why 60? Because spacer plus the repeats are about 30, so you have a multiple of 60. Whenever you get a integral multiple of 60, it tells you immediately how many fragments are acquired. So if you separate all of them, then you can clearly find out how many acquisition events have happened, right? So that is quickly you can do it, and that's what we have done it. So you can see that clearly there is an acquisition in the wild type, but when you delete either the alpha or the beta subunit, the acquisition is gone. We can also complement it against a deletion background, and the acquisition comes back, suggesting that the IHF could be a potential factor here. But what does this IHF does in the case of CRISPR acquisition is the next question. So, typically IHF binds to a very conserved motif. So, in contrast to other nuclear associated proteins, IHF has a very specific, it has a very specific binding. So it binds to this motif. So therefore, we scouted for the presence of these motifs in the leader region. And we could identify one. Lo and behold, yes, we do find such conserved motifs here. So there is a clear IHF binding site. So therefore, whatever we fished out seems to have some biological relevance. So when we carefully looked at the IHF binding site, it matched very well with the consensus. The next thing is, how do we test that whether it is actually involved in CRISPR? So you can do mutations. We have scrambled this site, we have done deletions and performed the same spacer acquisition assay that I have done. So you can see that only in the wild type, it basically, you see the acquisition, but in the others where you have mutations, it basically, there is no acquisition, suggesting that only the IHF is probably the host factor that we are looking for. What IHF does? It typically binds to the target site and it bends the DNA. So therefore, immediately we want to know whether when it binds to the leader sequence, does it also deform the DNA? So this you can actually do by using a fluorophore and a quencher. So we have labeled this IHF binding site that we have just predicted. One with the fluorophore, the other with the quencher. Whenever this IHF binds, it's going to deform the DNA, and that is going to bring these two together. The quencher is going to quench the fluorescence, and therefore that is an indication that it is in fact deformed the DNA. So when here when you add IHF to this reaction, you can immediately see that the fluorescence is getting quenched. But when you add a proteinase K, the fluorescence comes back, right? Suggesting that it is indeed it is basically bent. We also have actually measured how much is the bending, and it is very sharp. Right, so it is indeed creating a very sharp bending. So what we have so far found is that we found an IHF binding site in the leader, and binding of the IHF actually deforms the DNA. Right. So next question is, what does this deformation has to do with the CRISPR acquisition? Right. Okay, so that is the question here, and quite strangely, what we found was for the CRISPR acquisition to happen. The Cas1 and 2 basically has to bind to this site, right? So, we wanted to find the correlation between the IHF binding to the leader and the adaptation complex binding to the CRISPR repeat, right? So, this is basically the analysis we did. I'm just going to zoom it here. So, in the presence of IHF, you can see a complex, right? The mobility is retarded, so you can see this complex. But when you actually add the adaptation complex to this, you can see this band getting shifted up, right? Suggesting that there is a super complex that is forming, right? And suggesting that this happens only whenever there is an IHF. So you don't see if there is no IHF. Suggesting that the IHF binding and the deformation is probably required for the Cas adaptation complex binding to the CRISPR locus. So what does that basically means? How is it basically occurring, and why IHF binding has to be there for the adaptation complex to bind to the leader? That is the next question. So, so this is just to represent what we know of now. So IHF binds and bends, and when it bends, the adaptation complex is localized there. And why? So therefore, it appears that there is, in addition to the IHF binding site, there is something there in the leader region that recruits the adaptation complex. So, the leader region we have, this is the region that we have identified as an IHF binding site. And upstream of it, we just screened for whether any of these have any involvement in the adaptation complex recruitment. So we did the same mutation analysis, and lo and behold, you can see that when one of these mutation here, when you scramble this part of the sequence, you don't see an acquisition happening, suggesting that in addition to the IHF binding site, you have another site that is important for the integration. And this we call it as an integration anchoring site. And this anchoring site seems to be the one that is important for the adaptation complex recruitment. See, remember, the adaptation complex actually has to play the chemistry at the leader repeat junction. But why it also has to, it needs a binding site upstream of the leader repeat junction is something that we have to understand. So therefore, we have done the same EMSA assay with the mutant sequences. And you can see that whenever you have a mutant sequence, this complex is no longer forming, suggesting that this site is important for the recruitment. Okay. So when we actually closely looked at this upstream element, we could see another conserved site, and there is a highly conserved motif here, which is probably seems to be the recognition site for the adaptation complex. Okay, so now what we have basically found, to cut the long story short, is that whenever the phage infects the DNA, the phage gets fragmented, and the fragmented phages fragments are basically specifically recognized by the adaptation complex. It picks those fragments, even if it is longer, but if it contains a PAM element, it picks it up. Okay, so this is the PAM containing fragment, it picks it up. So whenever this longer fragment is picked, it only products a 33 nucleotide, a base pair fragment, and the additional fragments are basically pruned by this host exonucleases. And once this gets pruned, the IHF binds to this site, right? And it binds to the site and it deforms the DNA. Okay, and this is the site that we have identified, which is important for the recruitment of the adaptation complex. And you can see that this site is basically far away from the leader repeat junction where the action actually takes place. So it turns out that the recognition, the launching site for this adaptation complex is a bidentate site. A part of it lies at the leader repeat junction. A part of it is away from the junction. So therefore, in the one-dimensional space, these are far apart, right? So what IHF does is that it basically brings this closer in 3D space. And once it brings that, the bidentate site is regenerated for the adaptation complex. And therefore, now the adaptation complex can recognize this and the chemistry can take place. So that the 3' hydroxyl can actually initiate the nucleophilic attack, leading to the partial site integration. The other strand can initiate attack in the junction or in the distal end of the lipid. So now the space had got integrated here. So this integration leads to a gap here, right? So and that gap gets sealed by the repair enzymes. So now at the end of every integration event, you have a duplication of the repeat that happens, right? So this is the mechanism that we have addressed it with respect to this. This tells us how this specific integration actually happens, right? So so this is with respect to the mechanism of how the adaptation happens, right, in bacteria. I will just switch gear and run through the another interesting aspects, because what I'm trying to give you is a glimpse of functional diversity that we see in the CRISPR systems. So this is the another, the next part is the stage is the maturation stage, where the infection memory is retrieved, for which the pre-CRISPR transcript has to be processed and get assembled with the Cas proteins to form the effector complex. And if you look at again, the type 1 system is our playground. So here, if you look at this system, the 1E is the prototype. And here, this is how the conformation of the CRISPR RNA is. The CRISPR RNA is protected at the 3' end by this protein called Cas6. And it is basically coated by a multimer of Cas7. Because otherwise the RNA is floppy, so it has to basically have to be linear in order to base pair with the target, right? So on the 5' end is protected by this Cas5 protein, and you have this Cas8, which is important for the PAM recognition, and you have another another proteins called so this is Cas11. It has 2 subunits. So if you look at this multi-subunit assembly, it's like a right-handed filament, right? So this is the prototypical system, but the point to note here is that the Cas5 and the Cas6 are 2 ends of the spectrum, right? As I said, CRISPR always throws surprises, and if you look at one of these, here the Cas5 and Cas6 are fused, right? So therefore, with respect to this architecture, these two have to be far apart, but if it is fused, how this architecture is going to be different and how it affects the functionality is another interesting question. So this is the question that we want to address, and another talented student, Sunanda, took it up. So when we basically look at the system in Bifidobacterium animalis, typically the CRISPR array and the Cas operon are, either they have a single promoter or they can have a separate promoter. What I have shown is in the reverse strand. Okay, so this is on the reverse strand. Just for the sake of clarity, I'm showing it as a positive strand, but it is basically a reverse orientation. So you have this promoter here that transcribes this CRISPR, and this transcribes this. So this is how most of the CRISPR systems are, but there are exceptions, right? But in this, quite intriguingly, we also found another promoter that basically can, in principle, transcribe it from the other direction, right? So this is a system where you can transcribe this from the positive or forward direction as well as the reverse direction, right? So that intrigued us. The reason why is if it is made, if the RNA is made in this direction, this is how it is going to be. And if it, so just for the sake of clarity, we have named it as, so when the promoter is in the same orientation as this P-Cas, we call it as Cas-sense orientation. When it is in the anti-orientation, we call it as anti-sense orientation. So just to discriminate between these two. So when you have these two kinds of RNAs, the kind of repeat structures they are going to have is going to be different, right? So, so the next thing is when if indeed such promoter architecture is there, and whether both these RNA structures can be processed and mature to form the effector complex is the question. So when we basically looked at it, so we just looked at it whether it can be processed by this Cas nuclease. With respect to the CSO orientation, the cleavage occurs at the 3' end of the repeat. Okay, so therefore you have an 8 nucleotide 5' tag. But with respect to the CAO RNA, it happens at the 5' end of the repeat. So it appears that it can cleave both the forms of repeats, but the cleavage site varies. But apparently it appears to be variable. But if you look closely, the site is somehow have some conservation, right? So the cleavage site has some conservation. So therefore, it appears that it can process both orientations of the repeat, recognizing a common motif. So now, what is the implications of this? The implication is that if the CSO orientation is processed, the CRISPR RNA will have a repeat at the 5' end that will have 8 nucleotide 5' tag, and it will have a 3' tag at 28 nucleotide. So typically, all the CRISPR RNAs that are known today, most of them have the 8 nucleotide 5' tag. But if it gets processed in the anti-orientation, it will have a 5' tag which is longer than the 3' tag, which is unusual, right? Yeah, I'm done. So the next question is, okay, let's both of these are getting processed. Can they actually, form a complex to form the effector complex? So therefore, we expressed the CSO orientation is a normal one, but the unusual one is the CAO. So we basically co-expressed this CRISPR and the Cas, we reconstituted the complex, we purified them, we isolated and we verified that the Cas proteins are there. And when we isolated the RNA from here, it appeared to be closer to 70, which suggests that yes, it has an appropriate size. But when we did an RIP-seq, we also found that there is a 5' end processing that probably appears to happening, right? But the other parts of the repeats are in different stages of processing. It's not at mature completely. So therefore, this appears to be a possible scenario. The next is we also wanted to ensure so all these experiments we have done it in E. coli in a heterologous system. But the CRISPR system belongs to Bifidobacterium animalis. So we looked at the RNA-seq there to indeed verify whether both this kind of transcription happens from both the promoters. Indeed, we found that this is the forward strand, that's the CAO, and you can see that there is, it is getting expressed here as well as in the reverse orientation, suggesting that both forms of the RNA can basically form. And so the next is whether can it be functional as an effector complex. So this can be verified by an assay system in which you can integrate the CRISPR array inside the E. coli, and then express this cascade as through a plasmid bond copy, and the effector nuclease as a plasmid bond copy. And this is going to target a plasmid which has a complementary strand to one of the spaces here. So now, whenever the targeting occurs, what will happen is the colony is going to be less. So that indicates that the CRISPR is active. If the colony is more, that means that the CRISPR is ineffective. right? So this is sort of an assay system that you can have. So if you just test this in the absence of either if you use Cascade or the Cas3, you don't see the CRISPR active. But if you have all the components, it does recognize the target. This is in the sense, this is in the antisense orientation, and so is the case with respect to the sense orientation. So it appears that this system is very peculiar, that it can have transcription happening in both orientations, and both forms of the repeat RNAs are recognized and processed, and it assembles to form the functional effector complex. So this is very interesting with respect to the CRISPR systems, and this belongs to Bifidobacterium animalis, which is also a probiotic. So this is the Bifidobacterium has a very condensed genome. It is only 2 megabase pairs, and probably this is a strategy to pack things up. And here you can see that the advantage is that typically in most of the CRISPR systems, you also always see one directional transcription. But here it is two-way it is happening. Therefore, you can have the infection memory stored 2x times, right, in the same locus. So this is a very smart strategy by the bacterium. So this is what it is all about. So CRISPR, as I said, it evolves and comes up with surprises that allows us to appreciate the functional diversity within the system. And finally, I just want to leave you with this single message that in CRISPRs, there is a lineage-specific evolutionary plasticity that occurs that leads to functional versatility. Yeah, with this, I would like to acknowledge my group members and the funding agency. And I'd stop.
Thank you, Anand. Thank you for keeping on time, but we do have time for few questions. So
nice talk, Anand. So you said that the distilled part is
Okay, yeah. They are under relaxed. Yeah, all of they will be processed, but what I'm talking about at the genomic level, at the DNA level, right? At the DNA level, it is the genome is also evolved, right? So when there is no selection pressure, that part of the DNA can be degenerated. So the repeats. See, you also have to look at it in terms of the repeats are also very, it's a direct repeat. So you can have recombination occurring between them. So that event would basically collapse this memory, right? But it can only be selected if there is a requirement. So therefore, that selection pressure is more towards one end of the CRISPR array. That's what I'm saying.
Nice talk. For the first part of your talk, if you knock out that gene, can you do a plaque assay and show that the phage does not cause plaques?
Right. So you can do the same thing with phages also. So when we were doing, we were scared because we didn't use phage targeting. Reason being is that we don't want any contamination because we are a shared system. Working with phages have other side effects. So what we did was we did a plasmid challenge. So it's basically either phage or plasmids or mobile genetic elements. Both are basically targeted in the same manner. Oh, yeah. Right, it's a very good question. So I didn't touch that because, in the interest of time. Yes, the phages also have counter, a defense mechanism. So one way they actually evade this targeting is having mutations in the PAM site, which is basically important for the self versus non-self. That is one part. The other is it can also have evolved with very specialized system, as you rightly said, anti-CRISPR machinery that targets at different stages that I talked about. So phages also have an array of such anti-CRISPR proteins. Yes.
Anand, very nice talk. This is to your first part of the question in the continuation of phages. Some of the phages have been shown to have CRISPR system also, very small phage 5, right? So what are your thoughts about that? Whether they get this CRISPR system from bacteria or they infect it into the bacteria, or
Yeah, see, the phages also have CRISPR systems, yes, as you rightly said. Yeah, so that's also a counter defense strategy. When they infect the bacterium and they succeed, sometimes they can also pick up fragments, right? So that gets selected if that is going to provide an advantage. In that sense, if they have a guide RNA that allows the phage to succeed against the defense system, it gets selected. So yes, that kind of a selection is also happens.
if I can ask more. So my again to your first part of the question, first part of your talk, you mentioned about the base editing, right? So sometimes it is seen that the number of bases of the same cysteine are there. So which of the cysteine with this base editing would select for the conversion from C to U or C to I?
Which of the what?
So imagine you are trying to do a base editing from C to different base.
Yeah.
And within the adjacent site of this individual cytidine, you have another CCC units.
Oh yeah, yeah. The question, yeah, good. That's an interesting question. So as I mentioned that the Cas9, when it actually binds to the target, it opens up the DNA, right? And it covers most of the portion in that space. So you have a very limited space that is available, that is there as a single stranded region. That is a non-target strand, that is, I showed it on the top, right? So that limited space is where you have to fix your target site. Okay, so that is a narrow space. So that is why I said the base editing has, these are the restrictions because of which the prime editing has to be developed. So it is a challenge, yes. If it falls within that space, yes. Otherwise, you can't. Yeah.
All right. So in interest of time, let's thank Anand for that wonderful talk.
Thank you. Thank you for your time.